In our next session, we are fortunate to hear about cutting-edge activities related to COVID-19 vaccine safety surveillance in the United States. These activities are part of the U.S. Food and Drug Administration's Center for Biologics Evaluation and Research Surveillance Program, known as the Biologics Effectiveness and Safety Initiative. We will be hearing from two speakers in this session, Dr. John Seeger and Dr. Cindy Zhou. Dr. Seeger is a pharmacoepidemiologist and chief scientific officer for epidemiology at Optum, where he has been employed for more than 20 years. Dr. Seeger has conducted dozens of studies addressing regulatory drug safety questions across a range of drugs, vaccines, and disease conditions. Most of this work has involved the use of health insurance claims databases. And Dr. Seeger's methodologic expertise focuses on research issues encountered in these settings. Throughout his work, Dr. Seeger has remained keenly aware of the limitations of research using administrative data and has supplemented the platform of insurance claims with additional data where appropriate, including laboratory test results, surveys, medical record reviews, and has expanded into research involving electronic health records. Dr. Seeger is an adjunct assistant professor of epidemiology at the Harvard T.H. Chan School of Public Health. He is active within the International Society for Pharmacoepidemiology and is a past president and fellow of the society. Dr. Zhou is an epidemiologist in the Center for Biologics Evaluation and Research Surveillance Program at the U.S. Food and Drug Administration. She has been engaged in projects focused on the active surveillance for COVID-19 vaccine safety and effectiveness since the start of the pandemic. Prior to joining FDA, Dr. Zhou worked as a senior researcher at HealthCorps and was the principal investigator across a number of post-marketing safety studies for both drugs and biologics. Welcome, Dr. Seeger and Dr. Zhou.
Hello, I'm John Seeger, and I'm the Chief Scientific Officer for Epidemiology at Optum. And I'd like to share with you today some work that my colleagues and I have conducted on COVID vaccine surveillance. And this was conducted in conjunction with colleagues from the FDA under the BEST initiative. These are my disclosures. So to begin with, I'd like to think about what is needed for vaccine surveillance. And when you think about that, there are a number of different perspectives to consider, largely overlapping, but there's some unique features to each one. There's the perspective of the manufacturer, the government, academia, and then the population that might be at risk for safety outcomes after vaccination. Ideally, you'd want a system that has these features. It is able to provide accurate vaccination records along with accurate outcome identification. And between these two, you know who gets which vaccine and when, and you know what outcomes occur after they do get the vaccine. To help, you would also like to have an accurate enumeration of the population who receives the vaccine as well as who develops the outcomes. And you'd want to know about it quickly so that finding out a year from now about what was going on today may not be that helpful for vaccine safety surveillance. So the BEST initiative is one of a whole range of safety surveillance mechanisms that the FDA and CDC use to monitor the safety of vaccines. These include both active surveillance as well as passive surveillance approaches. And you can see that the BEST initiative is off to the right in the red box, and it's in the area that involves use of large linked databases to conduct the monitoring. So I'd like to tell you a little bit about Optum and how Optum fits in with the BEST initiative. Optum is a wholly owned subsidiary of United Health Group. And United Health Group is one of the largest companies in the United States with over 330,000 employees. United Health Group includes the health insurer United Health Care and also Optum, which provides health services as well as health services research. And Optum has a number of smaller companies within it including Optum Insight, which is the life sciences company of Optum, and Optum Epidemiology is part of Optum Insight Life Sciences. And you see a picture of our Boston office, which is the headquarters for Optum Epidemiology. There are also other parts of Optum, including Optum Health and Optum Rx. Within Optum, we have access to a range of data sources that we can use to conduct research. In this slide, I'm depicting on the left, the blue circles, the Optum claims data, and then on the right, the green circle is the Optum electronic medical record or electronic health record data. And you see the numbers in the circles represent the sizes of the data sources, and there's clearly some overlap between the EHR and the claims data that are also shown by the numbers. Now, focusing in on the Optum claims data, because that's what's going to be the subject of most of the presentation today, you see the geographic distribution of the Optum claims data. And this is one of the features about the Optum data that makes it useful for surveillance is that it has broad representation across the United States so that we can enumerate the populations under study and they provide generalizable results. So the large numbers for the Optum claims database that I showed you a couple of slides ago are longitudinal numbers. The relevant number for studying a new intervention, such as a vaccination, the introduction of a vaccination, are really the cross-sectional or annual numbers. And so that's what I'm depicted on this slide. So in 2019, we had about 18.9 million people in the Optum claims database. That's about 5.8% of the US population. And then that bar has a red portion, which is the commercial claims, and then the blue, which is Medicare Advantage. And so if you use just the red, the commercial, it's about a little over 4% of the US population. So the way we use the Optum claims data for conducting research is to create a timeline for a patient. And that timeline is bounded by enrollment dates. So first the patient is enrolled in the database, they become eligible for claims for healthcare services. And then at the far end of that line, they disenroll and are no longer eligible for claims. And in between enrollment and disenrollment, you will see claims accruing for the patient. And what you know about the patient is what you get from those claims. And so, for example, the claim, the first one that you see here, the HCFA 1500 form is submitted by a provider. And these HCFA 1500 forms are submitted by outpatient providers as well as laboratory testing facilities. And for example, you might see that COVID vaccine administration comes into the patient's record on one of these claims. And it also gives you other information about the patient's comorbidities or medications that were administered to the patient. Then you also see pharmacy claims. Most of these will be submitted electronically, but this is what a paper form would look like. And then we also see UB04 form, and these are submitted by hospitals. So you can see through this process, we can start constructing a patient's timeline. You see services, diagnosis codes, procedure codes that are associated with those services, and that you can start putting together a timeline of the patient that includes important milestones, such as when they received the vaccination and when they developed outcomes of interest. Whenever you're working with a data source, it's useful to know something about the data generation mechanism. And I showed on the previous slide a little bit with the different claims forms coming in, but those claims forms suggest the patient had an encounter with a healthcare provider, and the healthcare provider provides whatever services they provide, and then they submit a claim to the health insurer, and then the insurer adjudicates that claim. There's often a back and forth, and that's sort of depicted by the middle box to the right in red, showing the filing and adjudication of the claim. sort of the back and forth that there's adjustments made to the original claim that was submitted, and eventually it gets incorporated into a research database that we can use for doing the research. All of those steps are associated with some amount of time, and so they contribute to the lag between when the service actually happened, the patient saw the healthcare provider, and when it's available to us for doing the research. And I have at the bottom of the slide here some of the lag that we see. So for prescription claims, they are incorporated pretty rapidly, and we see them within a couple of months. And the same for laboratory tests and test results. But for outpatient services, there's a 2 to 4 months lag, and for inpatient services, there's 4 to 6 months lag. And these make these sorts of databases less suitable for active surveillance of vaccines. So in keeping with what I had outlined as the features for an ideal data system within which to monitor vaccine safety, we want to find a way to reduce the amount of time between the service and the time that would be available for us to conduct research on that service. And so what Optum has done is found a way to develop what we call the pre-adjudicated claims data. And basically this involves bypassing the last two steps from the previous slide so that the claims data are archived upon receipt and moved over into a research data set on a biweekly basis that reduces the data lag, not for the prescription or laboratory claims, but for the outpatient and inpatient claims, where the shortening of the lag is somewhere between 1 and 2 months. So once we have our pre-adjudicated database up and running, how do we use that pre-adjudicated database to monitor the safety of the COVID-19 vaccines? And this brings me to the rapid cycle analysis concept. What we do is we observe people who receive the COVID-19 vaccine, and then we also observe claims subsequent to the receipt of the vaccine that indicate they have a diagnosis or a procedure code associated with one of the outcomes that we're following. So first of all, you need to know which outcomes to track, and you'll see that in a later slide in the presentation. There's a set of outcomes that have been predefined for us to track. Once we have counted the occurrence of these adverse effects after vaccination, the question becomes, is that count above or below what you might expect if there were no association between the vaccination and that outcome? And this involves what we call background rates, sort of expected rates that we use the same population, maybe at a different time point, to find unvaccinated people, see how frequently they develop each of those adverse events that we're identifying. And of course, you'll need to follow good epidemiologic practices and account for the amount of time that they were under observation, also the age of the patients, the sex, and seasonality of any of the adverse events, along with anomalies such as the pandemic, because that will lead to sometimes lower, sometimes higher incidence of a range of outcomes. So this is the vaccines that we're identifying, and these are the codes we use for identifying those vaccines. And you can see these are CPT codes, administration codes, along with the NDCs that correspond to each of the different COVID vaccines, so that we can identify who got which of these vaccines, and also there's a date that corresponds to when the vaccination was received. Here is a visual depiction of outcome identification following vaccination for the rapid cycle analysis. First, there is an anchor date, which is the date of the vaccination, and then we look for the occurrence of an outcome, the adverse events of special interest, following that date. And that's depicted visually as the orange X with a superscript 2. So when that occurs, then there's an assessment of, is this a new event? And so you look back a certain amount of time, that's what's called the clean window, and you try to assess whether there's a previous occurrence of the same outcome. And in this case, the blue bar showing how far back to look does not observe any other outcome of the same type in that time frame. So then the X superscript 2 would count as an outcome event. Now, X superscript 1 is prior to the vaccination, and so it wouldn't count as an outcome of the vaccination. But if it, depending on when it occurs in history, it might be part of the background incidence rates to determine if the observed outcomes following vaccination are above or below or at expectation. And then you see also the X superscript 3 that occurs in the same risk window from following the vaccination, but is close enough to superscript 2 that it's sort of counts as the same event. And so those are not counted, X superscript 3. And then X superscript 4 happens after the risk window, which would also not be counted. So then what you do is you find all the patients who have an outcome of a particular type, you sum them up, and you also sum up the ones who don't have the outcomes. And this is all of your risk windows and the fraction of the risk windows or the counts of risk windows that have outcomes. This table shows the outcomes of the rapid cycle analysis, 16 different outcomes that we're looking for, 16 different adverse events of special interest. And you can see that they include things that you might expect following a vaccination, such as Guillain-Barré syndrome or anaphylaxis, and also deep vein thrombosis. It shows the setting within which we are looking for claims for the outcome and the time period, the clean window before which we'd be looking to see if there's evidence that there's a pre-existing condition, and then the risk window, different periods of time for which we will search for the outcome following the vaccination. And so, for example, things like anaphylaxis have a very short window of time after the vaccination. So patients could be counted as having each of these 16 different adverse events of special interest, but each one only once. Since the rapid cycle analysis is anchored on a vaccination date, we monitor the vaccine doses quite closely, and you can see we have a figure such as this that shows the time, calendar time on the x-axis and the number of vaccine doses by brand and by dose sequence. And so this is monitored closely, and this serves then as the starting point for the outcome identification. So once we've identified the vaccination dates and then identified adverse events of special interest in the time following vaccination, according to the parameters that I've just discussed, then it comes to a question of whether whatever you observe for each adverse event of special interest, is that above or below or at expectation? And to do this, we have to develop some expected incidence rates. And this is based on background epidemiology. We look at some period of time preceding the use of the vaccine, and we identify the occurrence of the same set of adverse events of special interest in an unvaccinated population. And then, of course, the results need to be presented broken out by age and sex, and that's what's shown here. This is not our work. This is from a European consortium that was doing essentially the same thing. Our work is still under publication at this point, but it will have a very similar look to it. And this is another way of looking at the background incidence rates that, beyond accounting for age and sex, it's also a question of accounting for either trends over time or seasonal patterns over time. And so this kind of plot showing the number of cases per 100,000 person years by month is a useful way to address this, so that you can identify things that you might need to account for in the analysis. All right, so once you've identified the vaccination, identified the outcomes, and then compare them to background incidents, then you're ready to present results. And here's some results of the Optum work. This was presented by Stephen Anderson in last month's International Society for Pharmacoepidemiology conference. And you can see the Optum pre-adjudicated claims data, ages 12 to 64, and the different vaccines, and the number of events after vaccination. This is for myocarditis, pericarditis, and then whether or not there's a safety signal observed. Now, there's a big step between the number of events that are counted and the determination of yes or no, whether there's a safety signal. It goes through all the process I just described of comparing to background rates, and it uses a particular statistical process called the maximized sequential probability ratio test, binomial version, and that test was designed for surveillance purposes to sort of provide a balance between sensitivity and specificity. And as data keeps accumulating, we may or may not achieve signals based on this process. But it's a statistical process that can be repeatedly examined as data accumulates. So I've described the rapid cycle analysis, sort of the primary surveillance activity that we're engaged in at this stage. But once we identify a signal, it might be time to move beyond the surveillance paradigm and go into something else. And that involves contextualizing the adverse event in relation to vaccines, as well as addressing competing hypotheses that may not have been a part or built into the surveillance itself. And so doing that is just a matter of good epidemiology practices. It really involves a customized study that would allow for inferences to be made about whether or not the vaccine increases whichever adverse event of special interest it seems to be related to. And that brings me to the end. I hope I have given you through this presentation some insight into how we are monitoring the safety of the COVID vaccines at Optum within the context of the FDA BEST initiative, and I will be happy to answer questions during the live Q&A session. Thank you.
Hi, everyone. Thanks, Professor Lee, for the invitation and introduction. Today, I will focus on some ongoing active surveillance activities led by the post-market surveillance team of FDA Center for Biologics Evaluation and Research, or Seeger, in the context of U.S. vaccine surveillance programs. I will start with an overview of the U.S. vaccine surveillance programs, followed by a brief introduction of FDA Seeger surveillance program, and end with some ongoing activities to monitor safety of COVID-19 vaccines. First, I will briefly introduce U.S. vaccine surveillance programs. The U.S. government has a responsibility for public safety with respect to vaccines. There are two pillars for post-market safety monitoring in the U.S., including passive and active surveillance. One passive surveillance approach is Vaccine Adverse Event Reporting System, or VAERS. It is a national spontaneous reporting system co-managed by CDC and FDA. VAERS can rapidly detect safety signals and can also detect rare adverse events. However, the main limitation is that we generally cannot access causality from VAERS data alone, but it serves as an early warning system for hypothesis generation. Besides passive surveillance, federal government agencies also maintain large and robust active surveillance monitoring programs, such as Vaccine Safety Data Link by CDC and Veterans Affairs Administrative Data System. Agencies also collaborate on analysis, and this U.S. government-wide approach provides advantages in covering a broad portion of the U.S. population. For example, FDA and Centers for Medicare and Medicaid Services, or CMS, have been leveraging Medicare data to monitor safety of seasonal influenza vaccines among population aged 65 years or older and those who reside in nursing homes. Next, I will briefly introduce Seeger and Seeger surveillance program. FDA Seeger regulates the following biologic products: including vaccines, as shown on the left of this slide. Seeger's mission is to ensure biologic product safety and effectiveness through active surveillance. And to serve this mission, the Seeger surveillance program is dedicated to create and use an effective national post-market surveillance system to provide data for evidence-based regulatory decisions. The Seeger Active Surveillance Program works with a diverse group of experts, such as epidemiologists, statisticians, data scientists, and clinicians to conduct active surveillance studies. There are three components of the Seeger surveillance program, including federal partners, such as CMS, academic partners, and the BEST initiative. And as you can see here, Optum is one of our collaborators within the BEST initiative. The BEST initiative has access to diverse types of data, including large administrative claims data, electronic health records or EHRs, and linked EHRs and claims. EHRs provide more granular clinical information such as lab results, and we will dive into the comparisons between claims and EHRs in the next slide. On average, data lag or processing delay for claims is about 3 months, but we are making efforts to gain access to special data, which is critical to response to the pandemic. For example, data lag of Optum's pre-adjudicated claims is about 2 months. Besides granular data, EHRs are almost real-time and cover both insured and uninsured population. However, the main limitation of EHRs is that they only cover snapshots of patients' clinical experience. In contrast, administrative claims data are excellent in tracking healthcare continuum, regardless of which healthcare systems are used. However, claims data suffer from long processing time or claims delay. Given the pros and cons of different data sources, we usually evaluate the fit for purpose at the beginning of each study. In the following slides, I will talk about some ongoing FDA active surveillance activities for safety of COVID-19 vaccines. We're currently using administrative claims data to actively monitor COVID-19 vaccine safety. Specifically, we have been engaging with 2 data systems. One is Medicare data through a federal partnership with CMS, and the other is commercial insurance claims data within the BEST initiative. The work that I am going to present today would not have been possible without a long-term collaboration that we have developed between Seeger and our federal partners and our private sector data partners. Seeger has been working with the CMS on safety and effectiveness monitoring of biologic products for more than a decade now. And there is a large body of publications for those who are interested. We have a little less experience with the private sector side, but have been working with them to develop this infrastructure that was needed when this public health emergency arrived. CMS Medicare data cover over 92% of US elderly, and this translates to approximately 34 million beneficiaries aged 65 years or older in the US. Medicare data can be linked to medical charts on the member level. And this capacity is important as claims are primarily used for reimbursement purpose and are subject to coding errors. Medical records help verify the presence of conditions. For near real-time surveillance, the BEST initiative uses 3 large commercial claims, including Optum, CVS Health, and HealthCore, to cover a large portion of the population younger than 65 years old, and to enhance the capacity to detect the rare vaccine adverse events. These 3 claims databases can also be linked to medical charts for case adjudication. The Seeger surveillance program uses near real-time surveillance or rapid psychoanalysis to actively monitor 16 possible adverse events of special interest, or AESIs. The analysis is stratified by vaccine brand, dose, health insurance system, and age group. To timely detect the statistical signal, we sequentially test cumulative data as they accrue in each database. And I wanted to mention that this method is designed as a screening method in order to monitor multiple potential adverse events and to quickly detect any large increase in risk following vaccination. It was used by US government agencies for H1N1 monitoring in 2009 and 2010, and has been routinely used by FDA and CDC for seasonal influenza vaccine monitoring. Here is a working list of 16 possible AESIs that were included in our rapid psychoanalysis. I wanted to begin by saying that there was no evidence of any causal relationship between any of these AESIs and COVID-19 vaccination when the vaccines were authorized by FDA. These AESIs were decided at the beginning of the surveillance period before the COVID-19 vaccines were available. And to select these potential AESIs, we considered serious events that have followed other immunizations, events potentially related to novel platforms and actual events related to vaccine failure or immunogenicity, or recommendations from other surveillance systems. You may have recognized that some of the AESIs have been identified or reported as safety signals, such as anaphylaxis, Guillain-Barré syndrome, thrombosis with thrombocytopenia syndrome, and myocarditis or pericarditis. However, robust epidemiologic studies are needed to verify these reported safety signals. As John presented earlier, in this rapid psychoanalysis, we compared observed counts of AESI after COVID-19 vaccination with expected counts using Poisson maximized sequential probability ratio test. The expected counts were estimated based on background rates of AESI in a historical cohort and standardized with age, sex, race, and nursing home residential status when it is feasible. Details about this method are available in our previous publications for those who are interested. In essence, what this method allows us to do is to establish how we are going to spend the alpha or significance level over the surveillance period that we are looking at. It establishes a threshold at each one of the times that we look at the data. If at one point the test statistic exceeds that predefined threshold, there is a statistical signal that requires further investigation. However, a statistical signal does not mean there is a causal relationship between the two, but it does mean that there is evidence that the observed rates are higher than the background rates, which in our case, we took from the historical cohort, and the statistic signal does require further monitoring. Here is a thumbnail summary of the 4 claims databases that we used for rapid psychoanalysis. The analyses are ongoing in CMS Medicare, Optum Pre-Adjudicated claims, and Health Score. It will start soon in CVS Health. The second column shows different update frequency given data sources have different processing time. And the third column shows the size of these databases covering different time periods. Based on billing codes such as CPT and NDC, we have accumulated approximately 21 million vaccine doses in CMS Medicare across 3 vaccine brands and 5 to 7 million in each of the commercial claims databases. Here are the results from the rapid psychoanalysis in CMS Medicare and Optum data sets. For anaphylaxis, we have seen signals in 5 of the rapid psychoanalysis that we are doing, and I highlighted in red here. We saw it for all 3 vaccines and in the CMS analysis, and for Pfizer and Moderna in the Optum analysis. This really was not much of a surprise. Anaphylaxis does happen after some vaccinations, and the COVID-19 vaccines are pretty reactive. The only other vaccine for which we've seen other signals in the rapid psychoanalysis are with the Pfizer vaccine in the CMS analysis. Those are for acute myocardial infarction disseminated into vascular coagulation, pulmonary embolism, and immune thrombocytopenia. And these signals have already been reported by the FDA, and we had a public statement this summer about these findings. It's important to put some context around these findings. So Pfizer vaccine was rolled out first in the United States, and the initial doses were targeted for some of those most high-risk individuals, including individuals in nursing homes and individuals with conditions that put them at high risk for severe COVID-19 outcomes. In the rapid psychoanalysis, we did not completely control for this potential confounding. So these statistical signals have to be interpreted with these limitations. Also, it's important to note that the elevated risk, which I'm not showing here, for these 4 AESIs were all below 2. And it is not very high by traditional epidemiological standards. We were able to detect these slightly elevated risks because these are relatively common outcomes, and we had a very large database to work with. And for the rest of AESIs, we either do not have seen a signal yet, or there have not been enough events that have accumulated for us to even start with the testing within the rapid psychoanalysis framework. To make sure that we were transparent and communicated these results following Pfizer vaccination, FDA issued a public announcement for those 4 safety signals in the CMS Medicare analysis. We have a screenshot of the web page on the left, where we posted this on July 12th, 2021. It says that we detected 4 potential adverse events following Pfizer vaccination in persons aged 65 years or older in the CMS data. I wanted to note that, again, we cannot establish the vaccine caused these events in the rapid psychoanalysis. Currently, we are conducting inferential studies in CMS Medicare data using standard epidemiologic study designs for better confounding control. Those results are not quite ready for dissemination yet, but we are reviewing those results that do a much better job for controlling for potential biases. And for those who are interested, we posted the study protocol on the BEST website. In addition to rapid psychoanalysis, the BEST initiative also conducts ad hoc analysis based on safety signals across agencies. So for example, Bayer's reported elevated observed versus expected analysis for myocarditis or pericarditis among younger males through age 49 and females through age 29. However, one of the limitations of Bayer's is that the population denominator is not really known, and therefore true rates of the incidence rates cannot be estimated. So we conducted another observed versus expected analysis for the messenger RNA COVID-19 vaccines in the Optum data. And this table shows observed unexpected case counts by vaccine brand, sex, and age group. As you can see in the table, for both vaccines, we have higher observed counts than expected counts within age group. And these differences are much larger in males compared with females. And I want to remind the audience that we do not have events for the 2 youngest age group for Moderna, as this vaccine has not been authorized to be used in these 2 age groups. I also wanted to note that these myocarditis or pericarditis events are rare in these young individuals. Therefore, we have not presented incidence rates. As you can imagine, those will be highly unstable. What we are doing now is to track event accrual within several of the commercial data sources within the BEST initiative in order to provide more stable risk estimates following the messenger RNA vaccination. So none of this is done by a single person. We have a wonderful team here at FDA, Seeger, and I want to particularly highlight Azadeh Shoaibi and Huili Wang, who have been leading the BEST initiative, and the rest of the Seeger surveillance program is listed here as well. And also, lastly, we couldn't do this without a strong collaboration with our federal partners and our BEST initiative partners. For those who are interested, a lot of COVID-19 active surveillance work gets posted on the BEST initiative website, and you can also get there directly following this QR code. With that, thank you for your attention.